Hi, I'm Karen Ocorr.
Hi, I'm Anthony Cammarato, and we're part of the Development and Aging Program at the Burnham Institute for Medical Research.
We're going to demonstrate our semi-automated heartbeat analysis program for understanding and quantifying heartbeat parameters in model systems with small hearts.
This is a high-speed movie of a beating fruit fly heart. Information about heart morphology and contractility can be obtained by measuring heart diameters during contraction and relaxation cycles. To do this, the edges of the heart need to be identified and marked during maximum diastole and systole. In the pre-process window, select the movie or movies you wish to look at. We will select the movie you just saw of a heart from a young fly. Optical recordings can be advanced at slow speeds and even one frame at a time to allow precise identification of the frames where maximal contraction and relaxation of the heart occur. Make one pair of marks to identify these edges. Advance the movie to a frame where the heart is maximally contracted. Make another pair of marks to identify the systolic edges. Marks can be made in duplicate at different horizontal locations along the heart. The heart diameters obtained here will be used to calculate the percent fractional shortening, which provides an estimate of the contractility of the heart. This measure